Hello, everyone. Today I will be presenting on this paper that was published by a research group at the Yale School of Forestry and Environmental Studies, and it is titled Biotic Interactions Mediate Soil Microbial Feedbacks to Climate Change. For some background information, this paper discusses land carbon climate feedback loops, which are driven by soil microbial decomposition. So this happens when microbes in the soil decompose organic material, and this process releases carbon into the atmosphere. These soil carbon dynamics are very sensitive to climate change, and they can be affected by both abiotic and biotic factors. Under climate change predictions, there will be greater nitrogen availability and warming temperatures in the soil, which could affect the microbial activity and the amount of carbon being released. And this paper looks at how biotic factors such as animal grazers may be able to control this increased microbial activity. Specifically, it looks at fungal cord growth and how these fungal cords may be controlled by isopods, which is this little bug shown here. So these isopods may be able to graze on fungal cords and be able to limit their microbial activity. So papers looking at these global change drivers should consider different trophic levels and not just look at surface level grazers, but primary consumers within the soil as well, such as these isopods. The study is significant in its relation to climate change. The Intergovernmental Panel on Climate Change considers these soil feedback loops because of their ability to release so much carbon into the atmosphere. And forest ecosystems contain about 40% of the global soil carbon pool, and decomposition um, results in about 7.5 to 9 times that of anthropogenic emissions for carbon annually, which is quite a lot. So any changes in these soil feedback loops due to climate change effects have the potential to exacerbate climate change if this microbial activity is being increased. And so this paper is looking at how increases in temperature and nitrogen availability are predicted to be able to enhance this microbial activity, which could be very problematic for carbon releases. So this paper has two hypotheses, the first being that global change drivers increase microbial biomass, and they're specifically looking at fungal cords as well as these global change drivers enhance microbial activity. Secondly, they're looking at how soil animals, specifically isopods, may be able to control microbial activity through grazing, and specifically grazing on these fungal cords. Their experimental design, they tested two global change factors, which were soil warming and nitrogen addition. And then they tested additionally for biotic treatment chambers, which were the presence and absence of isopods. And this figure, part A, shows their overall plot of land they were working with. Part B shows 4 separate biotic chambers. And then part C and D, C is a chamber that is lacking isopods, and D is a chamber that has isopods present. And you can see just visually the soil surface of these do look different. So for their results, they looked at how these abiotic treatments affected fungal biomass. We see the abiotic treatment on the x-axis. C is control, N is nitrogen, W is warming, and W plus N is warming plus nitrogen. And then the fungal biomass is on the y-axis. The different shades of gray are the different biotic treatments. And the letters at the top are if the results are statistically different from one another. So all the A's are similar and the B's are statistically different. So they found notably two significantly different results in the nitrogen and then the warming plus nitrogen abiotic treatments. And this was both for the biotic treatment, that is the presence of fungal cords, but the absence of isopods. And so they found that when these fungal cords have more nitrogen, which nitrogen is a limiting factor, so when there's a greater abundance of it, we see that these fungal cords are able to grow more. And then there's also no isopods present to control their growth. And so they also looked at the effect on enzymatic activity, that's in part A. And they found kind of varying results, but for the nitrogen and warming plus nitrogen treatments, again, they found a positive result for the biotic treatment of the presence of fungal cords, but the absence of isopods. So again, we see that not only is the fungal biomass increasing when there's more nitrogen available, but the microbial activity, enzymatic activity is increasing as well. And so they also looked at the effects on fungal mediated wood decomposition rates. So for this part B, fungal mediated mass loss just means the amount of wood that was decomposed by these microbes. And they found that, and they only tested these two biotic treatments here, which were the presence of cords and the absence of isopods, and then the presence of cords with isopods present. And they found for all four abiotic treatments that when fungal cords grow without isopods, they seem to have greater activity than when these isopods are present, which again shows that these isopods may be regulating the activity of the microbes. Next, they looked at, in part A, they were looking at fungal biomass on the x-axis and the amount of fungal biomass removed by isopod grazing on the y-axis. And you see when fungal biomass increases, so does the amount being eaten by those isopods. But it is not a direct relationship. It seems that as fungal biomass increases, the proportion of the fungi being eaten by the isopods also increases, which is kind of interesting. And then in part B, they're looking at the presence of nitrogen on the x-axis and fungal biomass on the y-axis. And we see in the blue, that is when isopods are present, and the red shows when isopods are absent. So we see that the blue line sort of stays steady, it's not increasing, but the red definitely is increasing. And again, this shows that when isopods are present, they control this biomass and activity through grazing. But when they're not present, there's a steady increase in that biomass. So, some main takeaways, these results were in favor of the hypotheses. The global change drivers, nitrogen addition, and soil warming do result in a net positive effect on microbial biomass, specifically fungal cords, as well as that microbial activity when isopods are not present. But when those isopods are present, they're able to graze, and this results in that top-down control where the biomass and activity of the microbes is actually being managed by them. So isopods usually graze on leaf litter, which is actually a stimulating behavior in terms of microbial decomposition. But as nitrogen becomes more available in the soil and fungal cords continue to grow because of that, this may cause a shift where isopods primarily graze on fungi, which this grazing is a limiting behavior in terms of microbial decomposition. So this could cause a significant shift in these soil feedback loops and result in this top-down regulation of these isopods controlling the mass and activity of these microbes. For studies that are testing global change drivers in the future, they should consider top-down effects of soil grazers, as this paper has shown, because they are significant. And if they aren't considering these effects of soil grazers, they may find that they have inflated results of these carbon cycle climate feedbacks and have higher predictions of the carbon that may be released by microbial activity. And forest management practices might consider harnessing these effects of the invertebrate grazers if they have a lot of fungal cords present in their soil. They can use these invertebrate grazers as methods for top-down control to avoid these carbon sink losses, and which would contribute to climate change. Here's the reference for this paper. Thank you for your time, and I'm happy to answer any questions that might come my way.